It is my great pleasure to introduce you Professor Didier Raoult from Marseille. I told you he was from Marseille. Told you he was such a powerful personality that he could make the train derail. That's correct. He just arrived, so be, nice to him. Didier Raoult is a friend for many years, but he's a very special person because he has done everything that you can do in a university. He's a medic, may not like that. He's a biologist. He's very good in evolutionary biology. He's a humanist. He was a lot of things, but two major. Dean of the Faculty of Medicine in Marseille, but also rector of the University of Marseille. How many students in Marseille?
At this time, 25,000.
25,000. We are 13,000. If you look at the CV is very complicated because you start on page 1 and you have the reference and everything and you end up at page 150, so we're not going to go through a CV, except to say that he's going to talk about paleomicrobiology, and the idea to choose somebody talking about that, and especially Didier, he's the only person who can do that, is that you go through history, you go through archaeology, you go through anthropology, and you go through science, biological science, and evolutionary biology. I think it's very interesting. He's the most enthusiastic person I ever knew in this particular domain and in medicine in general. So Didier, it's your turn.
Hello, thank you. It's always a pleasure to come in Lausanne and meet my friends here. I want to talk, the reason where I like to talk about paleomicrobiology, it's because I think it's a good lesson specifically that I give to youngsters in my place to explain that science is not only discovering things, it's fighting for this, it's resisting when people are against this, and that happens with us in microbiology. It's been a terrible story, but very interesting, and a lot of fight with this. So if you're not ready to fight, you're not ready to find something great. If you're not ready, just do small things. But if you want to do something big, first people will start to say it's not true, and when finally everybody's agreed to say it's true, they will say it's not new, which is basically that means that it's important. And if you're lucky, you can have the two reviewers that see your paper, one saying it's not true and the other one saying it's not new, and the editor fire your paper despite the fact that they contradict themselves. So this is fine. So infectious disease and outbreaks are probably been very, very important in all the human history, probably all the living history. I suspect that I disagree with Darwin. I suspect that things have not been slowly evolving for most fit organisms, but I suspect that people and animals and plants have been destroyed massively by outbreak and they disappeared, and some few by luck are still there, but not prepared to the next outbreak at all. So it's probably what's happened with horses, for example, in America. They all disappeared 10,000 years ago, and you cannot say that horses are not fit in America. It's stupid. Disaster. So that's probably was, and among infection, disaster was probably more important, the most important disaster that happens as far as we know. So there have been several outbreaks that have been described historically, including the Thucydid plague that changed history because Athens was defeated by Sparta. After this, the Antonine plague was probably the beginning of the fall of the Roman Empire, the definite fall of the European Empire. Roman Empire was the Justinian plague that ended the story of the Roman Empire in Europe. And then the Middle East plague that probably kills between 30 and 50% of the human population in Europe. And then after Columbus, there was outbreak of syphilis, and outbreak of eruptive fever that were quite well described by Fracastoro. And in the other, so we import probably typhus and syphilis, and we export to America measles and smallpox, and probably 90% of the Native Americans that died after the conquest died of these two diseases. So it was really killing more than the Spanish soldier of everything. That's the outbreaks. And war and lives, that's, I will focus on that. So we have investigated several focus, including in Douai, that's in the 18th century, that was the Spanish War, Napoleon, and World War I. So, and the Bolshevik Revolution also was probably associated with 20 million of deaths because of lice. And the Spanish flu that have killed a lot of people in 1918. These have been documented. So the problem of historic description is that many of the clinical description is not compatible with what we think. You may understand that, in fact, the description of the disease is cultural. It is like this. So if you see the description of the disease by Hippocrates, or if you see that by Fracastoro, or if you see that now, they don't look at the same thing. So they don't see the same thing. And this is very critical. And in my own life, I've seen things that appear to be very obvious when you look at this that have not been described for 20 years because nobody was looking at. So the way you look determines what you see. This is very critical. And then, because we live at different stage, in fact, we don't see the thing. But one thing is very clear, and this is bubo plague. And it has been very well described, even by Procopius, during the Justinian time, and you get plenty of pictures of the Renaissance where you can see bubo, and specifically Saint Sebastian was considered the saint of the plague, and because of the arrows that you receive, and plague was like an arrow. So you can see pictures of him showing the bubo, and there is no any other outbreak associated with bubo than plague. So this was very well described clinically. And then when you want to know if there is an outbreak, the anthropologists say, well, there is several things that they call the catastrophe burial sites. So it's not regular cemetery, it's something different. So you get plenty of bodies. You can see that these have been done in a hurry, they put together, and then very basic burial procedures. They're not religious, they put all together. And usually it's outside of the cemetery place, outside of the church area. So you can recognize in the Christian times that it's not normal. And the reason are the war itself, but for the war, if they are wounded, usually you get something on the bone that remains. So you can identify that these people have died because usually they're young men and they get bones lesion. They could die of famine, of course, of cold, and of epidemics. And it's hard to define, when you say it's not the weapons, it's hard to define just based on this, what was the reason. And what you can work on to do the thing is that you can work on frozen tissues. It's what have been done for the Spanish flu. There are other works that have been done on this. You can look on fixed tissues. You get fixed tissues since the end of the 19th century. You can work on, I'll show you one if you want. And you can work on mummified tissues. And you can work on what remained, if not bone and teeth. I've been involved quite early by a friend who got a theory that I don't believe at all. He believes that syphilis was in the Old World before Columbus. And we have a very nice description of syphilis. So, but the reason is that in some case, you get some bones with this kind of lesion, which are typical of syphilis. And he say, well, pathologically, it looks like syphilis. And please do a test to show that it is syphilis. So, because he was a friend and it was funny, we tried this and it doesn't work. It doesn't work, first I think that because it's not syphilis. And the second thing is that because the bone here, when we tried to work, it was full of inhibitors and of contaminants because it was outside. So that was our first study, never published that. But 2 or 3 years after, he was investigating a focus of plague in Marseille. Marseille has been nearly destroyed. 70% of the population died in 1720 because a boat come from the south with plague within, and they get the quarantine. They get the quarantine was not respected, and then the town was completely infested by this. And there was a cemetery here that was found just around. And very interestingly, they were treated in an hospital that now is a wonderful palace hotel in Marseille named Hôtel Dieu. And in this place, we get the record of all the bodies that were buried. So we get all what happens on this guy. So he came in my lab and he get a piece with just bone and schools and say, Didier, what can we do with this to show that it's plague? And then we get very lucky. But if you're not lucky anyway, you will die just getting out. So you need luck in science like everywhere. So, but we were very lucky because when he come here, I get at this time a dentist in my lab who was looking to get a subject of Master 2. And I don't know what to give it to him because he was doing half-time dentist in his own practice. And just he get 2 or 3 afternoons to work with us. So we have to use what he used to know. And I was scratching what can I do with him. And then he get with his cool, and I say, well, we're going to look in the teeth. Nobody had looked in the teeth before to do this. So it was just a joke to start with. And then I remember I called him in and they said, you're going to do la peste des dents. And I said, what am I talking about? And then we started on this. After this, it became science immediately. Then we got an experimental model with guinea pig infected by Coxiella boneti and Rickettsial connori, showing that you can find that in the teeth. And in fact, what happened is that the teeth was a very good idea because the dental pulp is a very, very good sample. In the teeth, there is a soft tissue which is exactly like the spleen. So it's full of blood. And when you die, of course, the level of contamination of this compared to bone is very low because there is just a small hole at the end. There is a contamination, but very low. And then when you come back and you find these teeth, you find a sample of blood that have been saved. So there is a little piece of blood which is dry, lyophilized, dry, that you can use to do PCR, for example. And this is what we did. So you need, so we use experimental model in guinea pig and cats to prove this. And then the dentist did that. And you need to do it, well, it's specific. Of course, we can learn this, but it takes quite time to be good enough to do that, and you use the dentist tool. So this is Gérard Aboudaram did that, and he's still working with us. So he's really using everything you can I'm sorry, the video is not coming. Well, too bad. What he do, he takes the skull, takes the mandibula, take a picture, select the teeth where you get the biggest pulp, and then cut that, and then you scratch that, and there is some powder where you can do PCR. And then you go to something that is a big part of our job. We're doing, I don't know, 400,000 PCR per year. So it's diagnostic tests. It's just diagnostic tests. And this is one of the reasons why we did not understand the questions that people had about the result of PCR, because it's basically the heart of our work. So doing diagnostic PCR, whatever the sample is our job. So it was quite simple for us. So we did that. And then we reported that. We reported that first. I was very lucky at this time, and this is, you need to get a good editor. And this time I've been incredibly lucky because probably the most clever of the infectious disease people at this time was Lederberg. Lederberg was a genius, was somebody fantastic. And we were lucky when we sent that to PNAS that Lederberg was the editor of this. And he was the editor of these 2 papers. And we have been very lucky because it was rejected by, interestingly, it was rejected by Science, but when we published that put the 2-page comments on the papers that have been rejected by Science when we published that in PNAS. So that was funny. Well, it was, so we did that, and we did that on this outbreak of plague in Marseille, but this outbreak of plague in Marseille is just one of the relapse of the big outbreak of the Middle Age. So it's the same story that's come back several times in Europe until the 18th century. So it was a very, very long outbreak with relapses. And then, when we start this, as I told you, for us it was very simple. We have never worked on plague. We didn't grow Yersinia pestis at this time. We do in silico PCR. We amplify that, sequence that. That's it. I mean, I cannot see any reason why we could get a mistake out of it. And specifically because when we do something like that, we never use positive control. And this is something that you may have in the brain because even some reviewers, I've seen that it eventually published in the New England. I was really surprised. To evaluate a positive result, you just need negative controls. To evaluate negative results, you need a positive control. But if you want just to control positive results, you don't need positive controls. And the problem with positive control is that it is a source of contamination. So if you don't have positive control, and you have never tested one thing, it's impossible that you get a contamination. So it has to be true. And this is what I call suicide PCR. Okay? Just to say, if you test one gene for the first time and never reuse it at all without positive control, I can't see how it could be a false positive. There is no way. Okay? And we did that. I will explain you why we want to reinforce that, because when we first produced that, what happens is that another team come and they were not either familiar with TIS or with PCR, but they find negative results. And then their conclusion was, it's not because we're wrong, it's because they're wrong, so it's a false positive. And the chance that you have a false positive in this condition compared to the chance that you have a false negative is very low. So very interestingly, but remember that, very interestingly, Gilbert claims that probably TIS is not a good sample. First, because they don't get a dentist, so they did a very special way of using TIS. And second, probably the Middle East plague was not plague, but something else. And it was really interesting. Remember the guy, Gilbert. And the reason they say it was not positive is that, well, DNA have been so degraded that in fact it's too small for PCR. So and then this was something that come from people working on ancient DNA in human beings, that the two first works that have been done have been contaminated. But of course, it's very easy when you're in the lab to contaminate with human DNA. It's much more difficult to contaminate with Yersinia pestis if you're not working with this. Okay. And then they make a generalization about the problems that they have, the reason why they need to get white chambers and so on, because they get human contamination in looking for humans. Okay. We have evaluated that recently, even because it's been very interesting. And then it comes with something else that was, and you know, things are coming back, is that people were saying, and now it's interesting because we're going back in this sphere, that people say, in fact, what happened in the Middle Age was not plague, but Ebola, or one hemorrhagic fever that kill everybody. And this will come back. And then people, you know, first there was one people of anthropology that get all his NSF grants based on this, based on demography, and that was interesting. And there were two big bestsellers at this time. They were selling hundreds of thousands of books everywhere just by fearing people. You know, people like to be feared, you know, and by history, like by pictures or, you know, TV series or whatever, you know. So, and then we were just coming, saying, well, it's plague. You know, we were not invested at all in that. In fact, we didn't go in plague by ourselves. We were driven by life to go to this, just because it was Marseille and we get this school and I get these students, that's it. You know? But now we were in the middle of the fight. And because of this, the association with the negative result of Gilbert, with this outbreak, people start to say, it's not true. And we start the work, and even now you can look at this if you can find, oh, we can miss this. It's hard to find, you know? But during all this time, you know, in pushing on Wikipedia and so on, say, no, it's not, it's still unexplained. And it was very interesting. And we try to say, well, look, it doesn't make sense. And even we say, well, look, it is, we have been looking back to the description of Procopius. We have been back to the description of the French doctor of the Pope. His name is Guy Chauliac. And he described, he was in Montpellier. This is the place where we get the second study, just at the end of the 14th century. And he described very well the disease, and he described the bubble. So we knew that was an outbreak with bubble. And there is not, I mean, the Black Death name come 1 century after the beginning of Black was saying, you know, terrible death. Doesn't mean that people get hemorrhage. So the name Black was pessimistic. It was not a description of the disease. The description of the disease was fever, outbreak, and buboes. So it was really interesting, and then, there was the BBC, they come to interview us. I say, well, look, it's just diagnostic. But the conclusion in all UK is that it's not plague. That's what's the end. So that was really interesting. And, well, we continue this work, and then, of course, we try to go a little bit further, and then come, for the first time, another study come using the same tool completely independently. We just see the paper coming from Germany, where they find that the Justinian plague was caused as well by Yersinia pestis. We find that in parallel. So they find the same thing, using the same strategies, the same primers. And after this, altogether, there was 17 papers, reporting, samples with Yersinia pestis. But until this time, even at this time, some anthropologists say it's not demonstrated. 17 papers published by different teams, Italian, German, 2 French teams, ours and another in Pasteur Institute, and the German one. So that was really interesting to see the power of the media against, the basic things that have been published by everybody. And so it is then, very interestingly, in 2011, another team by Poinard in Germany published, well, that's it. It's Yersinia pestis that caused the medieval plague. In the same PNAS journal, and they did not refer our 2 papers published 13 years before using the second technique. At the end, they say, well, these are much better than bone. That was interesting. This is a big thing. And then the same thing published, and of course they show that teeth are better, and the same thing published after that in Nature, 13 years after, that there is no single reference from us, or any of the 17 papers that have been published on the same subject, testing the same thing. So that's it. So that means that not only the reviewer can be bad, but sometimes they don't do their work. They don't want to do the work. And you can publish in what is supposed to be the best scientific journal without anybody checking if these have not been published before, and if you don't have to refer to that. So this is why I'm telling, if you're not ready to fight, you're not ready to do science. This is a fight. People are not more nice in these words than in any other words that you know. So, interestingly, this is another thing that we are doing now. We have been testing a focus of an outbreak, and we have now we are testing all the possible explanation by PCR, and we've tested something like 2,000 teeth, including one focus here, and then we didn't find that it was plague. It was a suspicion, and then we decide to do that by metagenomic sequencing. Then we sequence everything, and we have been able to find in one of these focus that we've tested negative before, we find sequence of Yersinia pestis. So we can find that doing PCRs that we have not done before to see if it was true in this focus, to explain why we didn't find that the first time and why we did find this one the second time. And the reason why that when we go back to the anthropologists, in fact, there were different foci that probably correspond to different episodes of different things. So just testing one focus doesn't mean that you explain the whole thing. So this was one interesting thing. So this is the last technique that we've used to do this. So now if you look, is it true that it's small? If you look at this is the size of the amplicons that have been amplified by different teams in all over the world. So some of these are quite big and some are small, but in fact, they are much more long. There is several reasons. First, if you kill a mammal, you die immediately, and DNA starts to. But if you put the bug in the soil, sometimes it can survive 300 years, and nobody knows. I can tell you for Yersinia pestis because we have put that in the sand and have done Balthazar and Moray for 2 years, and going back, it's still living. So you cannot believe that the DNA is degrading as rapidly in bacteria as in human beings. Probably not true at all. And even there is, very, very study on very, very old samples of bacteria where you can get very long DNA. So it's different. And of course, if they are resisting in the environment, like bacteria with spore, you can keep that probably for centuries. So we get some positive culture of amoeba and bacteria from the Middle Age, coprolites from the Middle Age, still living, because they get a cyst or a spore and survive with it. So you cannot say there is no, all the DNA disappeared. So as I told you, the bubble was well described. So we knew now that it is plague. Plague is plague. It's called by Yersinia pestis. But so why the epidemiology and the demographic explanation are not matching with the facts that we know now? There is something wrong. As I told you, this is San Sebastian, and you can see it's showing, the bubble here. So this is 16th century, so we know it's plague. And well, Gilbert is specialized in saying that everybody is wrong. So everybody gets specialty. So he says that what we say was wrong. He says that what the guy published in Egypt mummies was wrong, and even says that human beings didn't have genes from Neanderthal when he interprets things, all true is a few years ago. So he gets a very bad negative predictive value in general. So, and then, very interestingly, without any shame, the last description in Lancet Infectious Disease by Poinard, saying, well, we find that Yersinia pestis is also the cause of Justinian plague. He says, well, this is very interesting, and then he forgets that he claims that Yersinia pestis was not the cause of Middle Age plague. So this is really interesting. So you can see that it's very easy to see. Well, anyway, we try to understand now. We try to understand now. Let's go on this. Trying to understand what's happened and why the outbreak was not what you have been teaching. If somebody teaches you something on pestis, which is plague, which is not certain. But if somebody teaches you plague now, because nobody has worked on epidemiology for 70 years, when nobody is working, things are coming, more and more simple. You forget everything around. And finally, what do they teach? Probably what Philippe is teaching is, if he's teaching a plague, he's saying, well, it's easy, it's the rat, the flea, and the humans. And sometimes interhuman transmission in pneumonic plague. Probably you know more than this, and you don't say that. But this is what I would say 99% of teachers in microbiology and infectious disease will say. And it's not true. It's impossible. I'm agree with people saying demographically it's impossible. You can't imagine that the flea-transmitted disease will kill 70% of the population. It's stupid. It's not true. The flea bites the rats. They don't bite. They're quite specific, and it's only in very specific situations that they will bite you. So it's not true. It can initiate the outbreak, but it cannot cause the outbreak. So we start to look at this, and something happened. So we try to look at this, and our theory was that, so this is a major theory, so rodent from the outside, this is true, and then some rodent from the outside exchange flea with rodent from the town, the rat, and then the rat with its flea, the oriental flea, is transmitted, making an outbreak in rat, and some of these will bite the human beings, and the human beings will eventually transmit that by aerosol. In fact, we look at the lice as very old papers, and we are working on lice-transmitted disease, and in fact, as far as we know, the giant outbreaks that have been mainly caused by vector-transmitted disease, including by lice. Lice kill like mad. They kill by typhus, kill by recurrent fever. And then we suspect that lice may be a vector. And we start to work on lice. This Muku Uglu is the archaeologist of the lice. We work together to identify that. And then we test, is Yersinia pestis another louse-borne disease? Remember, the other one are Borrelia recurrentis, Rickettsia provazeki, Bartonella quintana. Okay, so we start to work on ancient lice as well, on this mummy, to try to see if there is a Bartonella quintana and which kind of lice they get. And then we go with Linda, we make experimental model in the lab. we can raise lice on rabbits. We infect the rabbits, then the lice are infected. We show that the lice are infected, and then we put this lice on another rabbit, and the rabbit die of plague. So a very simple model. It's a vector. So we find this. And then, what was this? And I suspect it was somewhere in my brain where I forget it, because it was the first conference that I've seen was done by Balthazar. And Balthazar spent 30 years of his life working on the epidemiology of plague. And what Balthazar found, Balthazar report, I will show you the paper. Balthazar found that in lice, in an outbreak in Morocco. And say probably, in fact, the outbreak of plague is caused by the human ectoparasites. And if you look at old books, and specifically if you look at Washington was writing how people should behave when they are in the Congress. You know, as I say, it's very interesting. And he say, you should not scratch yourself in public because everybody get ectoparasite at this time. Everybody get lice and fleas. Ectoparasites were extremely common. They start to disappear in the 19th century. So getting ectoparasites, that's the flea, Pulex irritans, and the lice was extremely common. As it is in poor country, it's very common. Everybody gets lice. So it's very, very common. So then if you realize that it could go in lice, and that lice could transmit that by the lice defecate on the skin, and then you scratch yourself and inoculate yourself, and then you get the disease. But of course, I get back and find this paper that show, you know, by Balthazar. What did he say? He say, well, the louse may transmitted Yersinia pestis. He finds that and shows that even at this time in experimental model. Of course, if you're doing PubMed, you're not going to find this. But if you look at Balthazar, you're going to find his family have put all his papers on Google, and you can read that and find out. So you do the bibliography if you know that it is Balthazar. And then to confirm, so this was theoretical, but to confirm this, what we did is that we start to look together to all the microorganisms. We find several times during this outbreak an association of Bartonella quintana, which is mainly transmitted by lice, and Yersinia pestis. So we have indirect evidence that some of these outbreaks were louse transmitted, and probably by the two together. So this was another one. And finally, we have somebody in Marseille working with in the east of Congo, which is one of the places where plague is still endemic. There is sporadic cases of, and it's an endemic place close to the Burundi and Rwanda. This is a very specific place. The two places where there is still a lot of cases, Madagascar and this place. So we organize a mission there to get some samples. So we find, not only this, we find plague in the lice, directly, currently, on the time hour. But even more interestingly, we find that in head lice. And head lice have not been associated with outbreaks so far. So this is first, now we know that Bartonella quintana can be transmitted by head lice. So it makes things even easier to be transmitted if you understand that head lice could be involved. Well, now, where is it? The other thing that say Balthazar was that the reservoir was not the rodents, the reservoir was the soil. It can survive in the soil for years. And then when rodents come in the soil and scratch the soil, sometimes they get infection and start a new outbreak among rodents. So, you know, it's very different of what we're talking about, you know, the rats, the fleas and the human. It's the soil, the rodents, and then the human, and then the human ectoparasites that makes the outbreak. And this is probably the reason why we don't have the outbreak now, except we can get that in place where lice are very common. And then all the things that have been described by Balthazar, we test that with modern tools and show, yes, you can get that for years. And even Michel, now, I believe that I've been the source of all this because I know the whole story, but Michel got a new data on Yersinia pestis that's going to explain the whole reservoir of Yersinia pestis in the world, but it's too early. Maybe you can ask Michel to come next year. The other one transmitted are Rickettsia provazeki, the agent of typhus. And one of the worst things that happened was during the Russian War of Napoleon. He go there with 50 to 700,000 soldiers. And you're concerned because there was a lot of Swiss guy with him. It was not only French. Remember, Swiss was part of France at this time. I'm sorry to say that. So the soldiers come from here, from Germany, from Italy. So that was the French army, but the France was a little different at this time, probably twice the size of what it is now. And it's probably one of the worst wars. So you can imagine, you get more 100,000 of people die during this campaign. Only 50,000 come back, and they die. They suffered and died mainly from cold, parasite, and fevers. And it was well described by Tolstoy when he describes this war in War and Peace. He describes that in the camp, people were putting the lice in the fire, and you can hear, the lice like tick, 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 when they explode in the fire. So it was described that all these people were completely full of lice. And this was a retreat from Moscow, and then there was the Berezina that remained, a name terrible for us, the Berezina, catastrophes. And just after there is Vilnius, and this was from here to here and to go back, they lose 90% of the people. And probably, 50,000 in the war and all the rest by fever, cold. And then typhus was suspected. And then I go back to the guy who gave me the school from Marseille and tell me, give me something from Russia, or give me something from Russia. So I want to show that it's typhus, because it's the beginning of my work is Rickettsia. So after a while, he invests a cemetery in Vilnius, just next to the Berezina. And when people invest that locally in Lithuania, they first believe that it was a camp from the last World War, Stalin. And in fact, there was hundreds of buttons, of uniform buttons from Napoleon's. So they were Napoleon soldiers. And in fact, if you look at the text, there was probably 30 or 40,000 people who died in Vilnius, not by the war, because of, famine, cold, and fever. And then we investigate that, and we test 30 human, and we try to obtain remnants of lice, because in the sand, and there was remnant of garment, and then we get a flotation. We test different flotation system, and the best one is kerosene. With the kerosene, the lice, dry lice with floats, and then we identify several lice where we can confine by PCR and find Bartonella quintana. We associate with this lice, and then we test these teeth, and we find that probably when they die, I'm not saying that they die of this, but when they die, 30% of them get bacteriemia with either Provazeki or Bartonella quintana. Well, we find typhus as well, and this is the oldest biological evidence of typhus in Douai during the Spanish War succession. And this is the site of Douai. And now Bartonella quintana. Bartonella quintana first described in the World War I, and this was named trench fever because they were described as having lice in the trench and fever. And this is now it's still common in human beings. So if you look at homeless here, in my hometown, there is around 800 to 1,000 homeless living in the street, and approximately 5 to 10% get bacteria circulating in the blood without apparent symptoms. Sometimes, probably after a while, they get endocarditis. But for years, they can keep Bartonella quintana without any symptoms. So it's like cats and Bartonella ensele that can get that for years without any symptoms. So it's a very adapted, very specific pathogen. And it is transmitted, we know that mainly, probably not exclusively, but mainly by body lice. And this is homeless with all the scratching lesion because of body lice. And we don't remove the teeth of one homeless to look for Bartonella quintana, but this guy was sick. The dentist wanted to remove the teeth, and then we asked him if we can test Bartonella quintana, and yes, we can find it, Bartonella quintana, in the teeth of this. So this is the only proof, not in animal model, but currently somebody having this disease and where we can find it. And then we were associated with the Henri Delumlé, who was the head of the Muséum d'Histoire Naturelle in Paris, and we say, can you give us old teeth to test that? And he gave us teeth, 4,000 years old teeth. And at this time, of course, everybody was plagued with all the lice that you can imagine. And in one case, we find Bartonella quintana, which is the oldest that we have found so far. So what remained for the Athens plague, the people in Greece want to test the different etiologies that have been put all the time, and finally they find that the focus that they test was typhoid. So probably, I don't know if it's all, because there is foci, all the case of the plague of Athens were typhoid, but it is possible. When you see the description by Tucid, it doesn't look like plague. There is no description of bubo, and the bubo, as I told you, everybody can say, well, there is a bubo. So if there is bubo, we may know that. And there is people who have been doing another technology that they absorb the antibodies and test the antibody from the bone and show that some of this bone come from people with syphilis in the pre-Columbian America. So this was the serodiagnostic tests of people before this. So, and there is another paleomicrobiology study which has been done in Egypt, where they found out that Tutankhamun, when he died, gets malaria, which is not, I mean, surprising, but it has not been demonstrated before. So maybe he didn't die of malaria, because in an endemic country, many people get malaria without any symptom, but when he died, he get that. Well, and this is a work that have been done recently in my lab by Christelle Denues. We work in coprolites. Coprolites are, you know, the stools that are very ancient. They become like stones, okay? And in some case, like in this case, it was a middle-aged coprolites that were in barrels. The barrels was completely closed, and then an anthropologist gave it to us, and then we take the middle of this to avoid contamination, like purification, and see what you can get, and mainly what you find. Not only she finds that by metagenomic, but also you can see by electronic microscopy. The main virus that she found were bacteriophages, but then you can study that as well. And she finds also a lot of parasites that have been identified currently, you know. So if the parasite has tear and tie, you can imagine, and we get amoebas that, of course, they get DNA. So that's the idea that after 300 years, there is only a small piece of DNA. It's not true. Well, cats and Bartonella. Cats are like humans with Quintana. Cats are harboring Bartonella encellae, but at much more high level. You know, 30 to 45% of the cats, specifically the young cats, have Bartonella encellae, and this, if they scratch you, they will give you cat scratch disease, which is caused by Bartonella encellae. So, and it's very common. This is red blood cells of the cat, and this is Bartonella encellae. Very, very common. And the same, we have been, the story starts when they rebuilt the Louvre, you know, there was an anthropologist, they find cats. And cats are quite late in the Roman didn't get cats, so it came lately. So the cat spreading come after the Roman Empire. But there were plenty of cats there, and then they send us some cat teeth to see if we can find Bartonella encellae, and we find Bartonella encellae. So we know that Bartonella encellae come from Europe. It was before Columbus time. And Michel Dancourt get an opinion on this. It's opinion. They say, you know, the king was curing the écrouelle, which is, you know, the bubo of Mycobacterium tuberculosis, by touching this. And he says that he was able to cure that because it was cat scratch disease that cure anyway alone. So probably not. So I will finish. So this is the cat teeth. And then the remaining question is that there are several organisms in Greece. Is the Antonine plague? We tried to get some samples. Was it caused by plague? We would like to get smallpox in Amerindian, and we really want to get the sweating sickness that caused a lot of deaths in UK in the 16th century, but we need some samples because we don't get any, and I'm trying to get that in England, and there is no theory about what is the bug that kill the people at this time. Thank you.
So thank you very much, Didier. I think it's absolutely great. There are several messages in here. The first question I have to the audience, anybody working in criminal sciences? Nobody in criminal sciences? Ah, yeah, so you have to go work with him a little bit. So this is the whole evolution of humanity that is much more plagued with death due to microbes, to what is surrounding us more than war. I have, before a question, I have two comments. The first is you heard that a certain point he said what Philip is teaching you is crap. Philip is me, of course, as a microbiologist. So when you start to teach crap, this is for your future career, then you move to the rectorate. And you're sure not to teach crap anymore. So any question from the audience? Yes.
The question is that, was it possible to get all these samples? Well, in fact, the people, there was a lot of people that were really interested in working this. So as I told you, we started with one anthropologist in Marseille. That was, he get his theory. I think that this theory is false, but he still believes it. But then he get chance to get this. But when I published the first one, at this time, I was, the second study that I did was, I was president of university, and I got another friend that was another president of university in Toulouse. He said, well, I love this work, and I want to do the same kind of work with you, and I just have a new anthropologist that came and investigate a burial site in Montpellier, that is the beginning of the Black Death in France. And say, do you want to do that with us? And the second paper was the only one due by two university presidents in exercise in France ever in PNAS. So we did that together. And then all the anthropologists that get these things, they start to be interested in working with us and send us and ask us to work with that, specifically Mrs. Castex in Bordeaux. And so we start to receive a lot of samples from Italy and France and so on. So a lot of people were interested since the beginning to understand what's happened, specifically to explain this. And we get, of course, we get plenty of other things to investigate now. And we're testing all other things that we were testing with, specifically now that we know that metagenomic will help us eventually to find a viral disease. There is things that we have not published yet because we don't even have a theory to explain that. So it's too bizarre. But well, of course, when you're doing broad spectrum things, you find something very unusual. So.
Thank you. So unexpected discoveries, unexpected networking and relationships. So that's what you are doing here. Many unexpected. I mean, you will encounter people that you did not expect to encounter. Any other burning question? Yes.
First, let me thank you for the overview you gave in this field. I'd like to go further in the future. Do you think that in 50 years or in 1 century, this study with the teeth bulb will work again, considering the human development and evolution according to the technology and elements in food that we are using daily? Thank you.
I'm not sure I understand the question, but I believe that in 50 years or even in 5,000 years, there still have blood in the teeth. So if there is blood in the teeth, you may be able to find which kind of microbe are in the blood when the people die, which is just what we did.
Is this the answer to your question?
There is a very, but I can rebound on another, there is a very nice paper, but I didn't report this, which has been done by this was also ending the controversy with the size of the thing. People, because it was people in the Pasteur Institute, Parasitology, did a work, very nice, published in Nature Genetics, I think it's 2 years ago, on the dental plaque, not plaque, the dental plaque, to compare the dental plaque in the Neolithic and now, 10,000 years, 5,000 years, and 19th century, 18th century. And this is answering partly to the question that you're saying. So And what he did very simple, didn't get all the questions that we have to fight for 12 years with stupid guys. He do 16S RNA amplification and sequencing, and that's it. He do deep sequencing. And what he finds is that the population of the microbe of the teeth, superficial, not the dental pulp, the plaque, change in the 19th century, abruptly. And this was associated, we've done a paper on this, and this was associated with the fact that we stopped to eat sugar, a lot of sugar. Sugar come only in the 19th century at high doses. It's come because of slavery in the West Indies, and then you get plenty of sugars. Before this, you get sugars just in oday on fruit. And very, very few, only very rich people get all sugar. So the teeth, the problem with the caries and so on, come in the 19th century. And there is a relationship when we look then at, with the anthropologists, the level of trouble, of all that you get in teeth, for example, in the Napoleon soldiers, compared to now, they get much less problems that we have, that our children have. Now it's changing because of survival and prevention, but what they get in the beginning of the 20th century. And this was associated with the change in the microbiota of the mouse.